Hi friends, and welcome to chapter 7.2 on transcription and gene expression. So transcription is part of what we call the central dogma of molecular biology. And by central dogma, we just mean really gosh darn important, okay? So the central dogma basically says something like this. All of your cellular functions and your traits are controlled by proteins. We know that DNA has the codes for making those proteins, but it's stuck in the nucleus, right? And proteins are synthesized on ribosomes. So we use this molecule called RNA to carry those codes from the nucleus to the ribosomes. Okay, so the molecular biology most important central idea looks something like this. DNA gets transcribed onto RNA, which gets translated into a protein. And it's that RNA that's kind of our middleman. By now you should already be somewhat familiar with the transcription process, okay? But just to review, it can generally be thought of as the process where we take a gene, a segment of DNA, and we unwind it, and we use that unwound section as a template to synthesize a complementary mRNA strand. In eukaryotes, that's going to happen in the nucleus, okay? It's not during any part of mitosis, has nothing to do with that, okay? This is just general cell function. So we're in the nucleus, there's a nuclear membrane there. And RNA polymerase is that enzyme right here that does double duty. It splits the strands apart, and it also helps with the synthesis of the mRNA strand. Notice that the mRNA codes are complementary to the strand that it's using as a template. Now, one of the unique features of DNA is that these strands are complementary. They are not identical. So what do I mean? Well, on one strand, we have one set of codes, and on the other strand, we have a different set of codes. They are not identical. Again, they're complementary. So although the base pairs are complementary, if we were to use one strand to make a set of amino acids, that would be an entirely different set of amino acids than if we use the other strand. So for example, this DNA triplet says T, okay, come on stylus pen, T, T, C. If I were to transcribe that onto mRNA, that would be transcribed into A, G. Okay, don't know what's going on with my stylus pen here. It's on strike. Perfect. Good timing for that to happen. And if I translated AAG, so AAG into amino acid, I would get lysine. Okay, on the other strand here, AAG would get transcribed into UUA, and UUA would make phenylalanine. I have no desire to spell that out, sorry. All right, so again, depending on which strand I'm looking at, okay, I could use those complementary codes to make two entirely different amino acids. So this strand would code for an entirely different set of amino acids than this strand. So how do we know which strand to use, right? Okay, well, these double strands of DNA can be thought of sense and antisense strands. The strand that actually carries the DNA genetic code for making a particular protein is called a sense strand. Okay, so the sense strand actually has the codes that we need. The other strand is called the antisense strand. So I know it seems to make logical sense to think that, oh, well, since this one has the codes, this must be the one that I need to use as my template. Well, if you're thinking that, sorry for your life, but you're wrong. Okay, so let's say this sense strand had the codes A, A, T, and I really needed that to make the proper amino acid. Well, if I used that sense strand to make the mRNA would say UUA, and it wouldn't be the same. On the other hand, that antisense strand, its complementary codes would read T, T, A. And if I used that strand to make an mRNA molecule, it would read A, A, U. Well, now that's great because now this mRNA has the same codes that I had in the beginning. Obviously, U here has been substituted for T. But what I'm hoping that you're picking up on is that if I use the antisense strand to actually make the mRNA, then the mRNA will have the same codes as my original DNA that actually codes for the proteins. Okay, so that's kind of the trick there with complementary base pairing. I got to use the antisense strand to make the mRNA so that I can be sure that the mRNA has the original codes that I needed in my DNA. So again, the antisense strand is used for transcription because then the codes that are on the new mRNA are going to be the same as the sense strand. So what does this look like if we draw it? Okay, well, I'm going to start with something like this. So here's my double helix, right? I'm not going to draw in all the base pairs. When the double helix is split by RNA polymerase, it's split into two single strands. Now, I know when we talked about chapter 7.1 about the leading strand and the lagging strand, be careful. That has nothing to do with transcription. Okay, leading and lagging is only for replication. Here we don't give a hoot about leading and lagging. Here we're interested in sense. Holy cannoli, what's happening with my stylus pen today? Sense and anti-sense strands. Okay, sense strands contain the codes. Antisense strands are the ones that are actually transcribed. Okay, again, we're using the antisense strand for transcription because then the mRNA, which is complementary to the antisense strand, will have the same codes as the sense strand, which has the codes to begin with. Okay, so you're smart enough to know which strand is which, but how does RNA polymerase know which strand is the sense and which strand is the antisense? Well, remember, then we got to go back to chapter 7.1 and really think about DNA. So out of all of our 3 billion base pairs, very few of them are actually genes, actual segments that are used to make polypeptides. The other ones can have several functions, okay, like structural, or in this case, being a promoter or a terminator. Promoter genes are an example of non-coding DNA. They do not have a gene property. They're not there to make a polypeptide. That doesn't mean they don't have codes. Of course, they're made out of base pairs. It just means that they're not there to actually make a polypeptide. The promoter genes are there to determine, to help RNA polymerase recognize which one is the sense and antisense strand. When those parent strands are separated, okay, a promoter gene or a promoter segment of DNA is going to help RNA polymerase identify which one is sense and antisense. Now let's think about this. Am I going to find the promoter on the sense or antisense strand? Well, think about it. The promoter is going to be on the strand that's actually getting transcribed. So we know that the AntiSEnse strand is the one that gets transcribed. And so that's going to be the one where I'm going to find that promoter piece so that RNA polymerase can figure out where it should start the transcription process. All right, so the promoter is here at the beginning, okay? Then we have the actual coding sequence. How does RNA polymerase know where to stop? We don't want it continuing to transcribe the whole DNA molecule. Well, at the end of the gene is a DNA segment. Again, it's non-coding, called the Terminator. And lucky for you, it sounds exactly like what it does. It terminates or ends the transcription process. So on either sides of the actual gene, we have the promoter and we have the terminator. This tells RNA polymerase where to start, and the terminator tells it where to stop. Now, before we get into any of these crazy, insane details, let's just do a quick review of transcription from a previous chapter. We know transcription happens in the nucleus because I'm using DNA and I'm talking eukaryotes. RNA polymerase does double duty. It splits the strands and then it uses the codes, okay, on the antisense strand to make a complementary mRNA molecule. It's going to start where the promoter is and it's going to end where the terminator is. So in the beginning, we're starting with DNA and its codes are in triplets, and we're going to end up with a molecule of mRNA, okay, and we call those groups of 3 bases codons. Okay, so if this is seeming easy and, nice to understand, great, that's now over, okay? Let's get into the nitty-gritty of what's actually happening here. So again, when you're ever asked a question about what direction does this happen in and you're not sure of the answer, just put 5 to 3 because that's probably the right answer. But let's take a minute to figure out why, okay? So the direction of transcription, if we're talking about the gene, it's always going to move from promoter to terminator because promoter tells you where to start, terminator tells you where to end. In terms of direction, let's take a closer look at this diagram down here. Okay, so here's my parent strand of DNA. Here's my growing strand of mRNA. Notice that they're anti-parallel, just like DNA is. Okay, so when I'm adding a new base, that's going to be added on to this 3' end. Remember, 5' is the pointy end of the house, 3' is the bottom. So we're going from a 5 to 3 direction. So new nucleotides are always going to be added onto the 3' end. So we're going from 5' to 3' in terms of the RNA. Now, that's going to be different if you're thinking about things in terms of the DNA strand. In terms of the DNA strand, we'd be looking at the 3' and it's moving, that whole process is moving towards the 5' end. But I want your brain to be focused on the mRNA going from 5' to 3'. So here's how it's growing. This has already been made. This is in the process of being made. Of course, this is an active process. So it requires an enzyme. We already know that that's RNA polymerase, but it also requires energy. And I know I've trained you to always just think ATP, but here we have another source of energy. It's like ATP. Again, those are our NTPs. So we know about DNA NTPs or DNTPs. RNA, it's the same process, okay? So these nucleotides, actually, when they're free nucleotides, are NTPs. They have this second and third phosphate group attached. When RNA polymerase is trying to make this covalent bond here, it's going to need energy. It's an anabolic condensation reaction. And to do that, RNA polymerase is going to cleave or cut off these 2 phosphate groups, harness that energy, and use that energy to form this bond. So let's see if we can put this all into one big drawing here. I'm going to start with a small section of DNA that hasn't been unwound yet. if I can get my stylus to cooperate. And then we have a section that's unwound into its two strands. Okay. One of these is going to be the sense strand. It contains the codes. And the other is going to be the AntiSEnse strand. Okay, I'm sorry for all the noise. This little naughty creature is bringing in all the leaves and chewing them up. Great, so we'll enjoy that addition to our video. Lovely. All right, let's get back to talking about these strands. So we have the sense and we have the AntiSEnse strand. And RNA polymerase is going to know when to start coding or start transcribing when it reaches the promoter. And it's going to know it's finished when it gets to the terminator. Awesome. Okay, so now let's see if we can get some mRNA in here, shall we? Okay, so my mRNA is going to look something like this. Okay, it's already been to this promoter gene. Okay, so that means it's probably picking up somewhere along in here. Here's my mRNA. Here's my 5' end. Here's my 3' end. Okay, and I can tell that this process is going in this direction. Okay, for a couple of reasons. One, because it's going to start at the promoter and work its way to the terminator, also from the 5' to 3' end. All right, so I hope you're getting this. I hope you're feeling confident. Great, because we're about to add more stuff here, okay? So once we have a piece of mRNA, it's actually not done yet, okay? We're not done with it. This is what we call pre-mRNA. It's still in the nucleus, okay? We've transcribed an entire segment of DNA, a whole gene, but we need to edit it, okay? So here we're going to go through what's called post-transcription modification or post-transcription editing, okay? So to do this, we're going to get rid of certain sections of DNA, or not, sorry, not DNA, of mRNA called introns, okay? So introns are what leave. We don't need them. They are removed. And I know that this is kind of like backwards thinking, right? You would think that whatever stays in the mRNA would be an intron, but it just doesn't work like that. I remember this one by thinking about introns have to stay in the nucleus. Okay, they're not allowed to leave. See, remember that the nucleus, okay, kind of surrounds the DNA and mRNA, and then the whole goal of having mRNA to begin with is to leave the nucleus and go to the ribosome to be translated. Well, these introns are removed. They stay in the nucleus. They don't get to leave with the rest of the mRNA. And so then that leaves us with exons. Exons are the sections of the mRNA that stay part of the strand. Okay, so these are my keepers, and we keep them and we kind of glue them all together, and they are able to exit the nucleus, go to the ribosome, and be translated. Okay, so once we have taken these introns and cut them out, we have to then glue these exons together or splice them together. You may have heard of that in terms of like wire splicing, bringing two bins together. Okay, so to do that, we use these things called spliceosomes. That's a real word, spliceosomes. okay, and they're a special type of snRNA, okay, and we call them SNRPs. It doesn't matter, okay, you don't need to know SNRPs really, you need to know spliceosomes, help to recognize these introns, get them out, and then splice those exons into shape. Once all of our exons are kind of glued together, we're going to do one more piece of modification before it's ready to leave the nucleus, before it's considered to be mature. And that's, we're going to add on a cap and we're going to add a tail. So we call this cap the 5' cap because it goes on the 5' end of the mRNA. And we add a poly A tail to the 3' end. Poly meaning many, A, literally many A base sequences. Okay, and here's the tail. So we have a 5' cap, we have a 3' tail, we have all of our spliced exons together. This is what we now consider to be our functional or mature mRNA, and it's ready to leave the nucleus and be translated. Now, I'm really about to blow your mind here, okay? Because these exons that we have don't necessarily have to be spliced together in the same sequence. Those spliceosomes have the ability to kind of rearrange them if necessary. So I could slice together those exons in different combinations or different sequences. And this is one of the reasons why the same gene can make many different versions of proteins. Okay, so humans have only a fraction of the number of genes as a rice plant. How can rice have more genes than humans when humans are seemingly much more complex? Well, that's because some of our genes can do multiple versions of proteins. So the same gene can make different versions of proteins if I kind of glue these exons together in different combinations. All right, so let's do this drawing, okay? So I'm going to start out with a piece of pre-mRNA. And I'm going to separate it into a few sections, okay? I'm going to label these introns and exons. I know that after post-transcriptional modification, only my exons are going to be left. Okay, remember that's the job of the spliceosomes. And onto these exons, I'm going to add what's called a 5' cap, because that's the 5' end. And I'm going to add in a 3' poly A tail. Okay, so in terms of annotating with functions, so here are my mRNA codons. Those are contained within the exons. So I guess that's this whole thing. Okay, and these guys, so this, I don't know where you have room for this. I'm out of room here. Gosh darn it. These guys, the 3' poly A tail and the 5' cap, are there to protect the mRNA as it moves throughout the cytoplasm. So this sucker has to go from the nucleus through to the cytoplasm into a ribosome, and those are there to protect the valuable codes held within the exons. Okay, So we know that the whole goal of producing this mRNA is to build a protein, right? And proteins are actively involved in what we call gene expression. So gene expression is the process where genes are actively transcribed into mRNA and translated into a protein so that that amino acid chain can be folded into a functional protein and it can have some kind of function within a cell. Like I said, transport, enzymes, junctions, cell-to-cell recognition, all kinds of things. Now, why do cells need to express different genes? Why don't they express all the genes they have? Well, remember, all cells have a complete copy of the DNA. But what makes some cells different from other cells is the fact that they only express the genes that are important to their functions. And again, we call that cell differentiation. It's one of the things that happens during development of a multicellular organism. So this gene expression is really important. We don't want your muscle cells to start growing microvilli that we're going to find on your intestinal cells. We don't want them to express genes that they don't need. So how do they prevent that from happening? Well, there's a couple of different ways. The 3 types of gene expression that we're going to talk about are methylation, proteins, and the environment. Okay, can you see this picture down at the bottom? This one. What do these look like to you? These things. If you said nucleosomes, then you're my new favorite student, okay? So this blue ball here should be our 8 histone proteins, and then this black stuff is the DNA wrapped around each one of it twice, okay? So when we were learning about these things, I told you that they had a role in gene expression, but we didn't really go into much detail about that. Well, let's roll, okay? So these histone proteins can kind of help regulate these which genes get expressed. And one of the ways that they can do that is by attaching what we call a methyl group. Methyl, you may remember that from our discussion on fatty acids, is a CH3 group. And what that methyl group does is it kind of closes this system up and it prevents these genes on these segments of DNA from being transcribed. Okay, so that methyl group is like a blocker. It doesn't allow the transcription enzymes like RNA polymerase to access these genes. Okay, so if it can't access those genes, they're not going to get transcribed and they're not ever going to be made into a protein to express that trait. So this is why males can have an X chromosome, but they don't necessarily have all those characteristics on an X chromosome because some of those X chromosome genes are methylated and they're not expressed. Okay, so one of the other ways that we can kind of put the kibosh on gene expression is by having a protein bind to the promoter. Okay, so remember, here's our promoter region. Okay, that's the region of DNA that tells the RNA polymerase where to start transcribing. If we attach a protein to this promoter region that is a blocker, then it's going to block that transcription process. And even if RNA polymerase recognizes this as a promoter region, it won't be able to carry out that transcription. Well, the other way around is that we can actually attach a protein to the promoter region that acts as like a signal to RNA polymerase of like, hey, let's do this. Let's get this job done. We need this protein now. And examples of those are like some hormones, okay? So our steroid hormones, like estrogen or testosterone, what they do is they actually enter the nucleus of a cell and they bind to the promoter region of a gene and they tell RNA polymerase to hurry the heck up. Okay, they're like a signal of like saying, hey, we've got a promoter over here, let's roll. We need to produce whatever those things produce. So we need sperm, or we need a thickened lining in our uterus, okay? Whatever it is we need, okay, they bind here and they start to initiate that transcription process. And then the last thing that can affect gene expression is the environment. So when we think about our phenotype, how we actually look, that is a direct result of our genes and our genes' interaction with the environment. It's also one of the reasons why identical twins can often have different characteristics. They have identical DNA, but those genes have interacted differently with their environment. Okay, so what we're seeing a lot of here, lots of uses in agriculture. So I can actually express different genes by putting things at different temperatures. Some reptiles are like that in terms of gender determination. Like if the mother sits on the eggs for longer and makes them hotter, they can become females, where if she doesn't, they can become males. We're also seeing a lot of this in humans, where things such as pollution can actually turn on genes. People that live in urban areas with more air pollution tend to have hundreds of times more active genes in the DNA of their lung cells, which may be producing mucus and other things to try to counteract that. So the environment is definitely an important factor here. Okay, and that'll do it for chapter 7.2.